Well, now we're going to talk about cell theory. We're going to kind of build on the last chapter, which was all about molecules, and we'll see what molecules build themselves into. But first, we need to talk about the history of cell theory in this unit. The current theory of cell biology is that all living things are made of cells. This is sort of a foundational concept in biology. We don't think there are any living things on this planet that aren't made of little bitty living things that makes the organism work. Uh, Cells are the smallest living things on Earth. If you rip a cell apart, the stuff inside is no longer alive. But on the other hand, you can take cells out of living things and grow them in a culture dish. You may have heard of uh, like cancer cells being cultured or bacterial cells being cultured. You can take stem cells from a human and culture them and grow skin to put on a burn victim. But if you take a cell apart and you take the pieces out of the cell, they don't grow because they're not alive. So cells were discovered when the first compound lenses were being produced in the late 1500s and early 1600s. People began to look at, really small stuff. The first scientist we have a record of doing this was Anton von Leeuwenhoek. In the 1600s, he basically is credited potentially with inventing the microscope. Uh, Beyond a mere toy, he was the first to use stains to make things easier to see. He was the first scientist to see tiny single-celled organisms from pond water samples, and so he is credited with the discoverer of cells. This is a replica of Leeuwenhoek's simple microscope. There's a lens here. Specimen on the tip of this little needle here would be examined, and I'm guessing you'd have to look from the backside through the lens to see the specimen. The screw here was designed to put the specimen in the exact position so you could get a good uh, position on it to get a good look at it. But no one knew really uh, what to make of all these little tiny things they were looking at. Robert Hooke, an English scientist in 1665, is the first scientist to coin the term cell after looking at cork cells that look like this. He called all the tiny little compartments in the cork cells, and that name stuck. So this is a replica of Hooke's microscope. It looks more like a traditional microscope where you're looking through an eyepiece and you have an objective and light is reflecting up through the lenses. So cell biology didn't begin till the 1600s. Before that, we just thought you were alive because you were alive. After that, it started to become apparent that cells had something to do with being alive. Almost 200 years goes by, and finally, cell biology is really getting underway. A scientist named Robert Brown finally named the nucleus, which is visible in all eukaryotic cells, but not prokaryotic cells. Talk more about those in a minute. And then between 1838 and 1839, Matthias Schleiden, who was a botanist, and Theodor Schwann, who was a physiologist, developed the first parts of the cell theory, which we hold as a foundational concept in biology today. The concept is that all plants and animals, the plant part with Schleiden and the animal part with Schwann, are made of cells. Okay, and cells are the basis of the structure and the function of those plants and animals. So those were the first two parts. Another almost 20 years goes by, and a final statement is added by Rudolf Virchow in 1855 that new cells are only produced from existing cells, and that was a reflection of the growing belief by scientists like Louis Pasteur that spontaneous generation didn't exist, and that cells made cells. They didn't just show up from gravy broth or dirt or something. So worms made worms. Worms are made of cells. Only worms can make worm cells. So those ideas all combined are what we call cell theory today, which is all living things are based on cells. Organisms are limited to what their cells can do, and cells can only make more cells by reproducing themselves. Okay, so that's cell theory. That's a foundational concept. It's the foundation of medicine. It's the foundation of biology. Now, before we move any further forward, we got to explain thoroughly the difference between prokaryotes and eukaryotes, because now this is very appropriate. We're eukaryotes, being humans. Eukaryotes have their DNA within a protective organelle or structure called the nucleus. And we have, in our group, in the eukaryote group, we have some unicellular forms and lots of multicellular forms. So in this diagram, this artist, has drawn a nucleus. This is the nucleus. It's got big pores in the membrane around it. This is endoplasmic reticulum. This is a Golgi apparatus, and this is the cell membrane. So eukaryotes don't just have a nucleus around their chromosomes. They have all kinds of other organelles, which are these membrane things floating around inside of their cells that do things. Now that contrasts with prokaryotes, which really don't even have a chromosome. A chromosome is a stick of DNA. Instead, prokaryotes have like a hula hoop of DNA, and it's not contained in a protective membrane. This is the hula hoop of DNA that's sort of all wadded up, and there are no complex organelles floating around in the cytoplasm. There's no membrane-bound organelles in the cytoplasm around this the DNA. This group contains unicellular forms only. So while the eukaryotes include unicellular and multicellular forms, the prokaryotes include only unicellular forms. So the prokaryotes, I tried to tell you before, these guys rule the world. There's more prokaryotic material on this planet than there is eukaryotic material, but we don't see these life forms because they're so small. We see these kinds of life forms because they're so big. That's the birds and the bugs and the frogs and trees and bushes and stuff. They're all eukaryotes. Now, there's been a great debate on, where did the eukaryotes come from? The first life on Earth, the fossil record shows, was probably prokaryotic. So where did the eukaryotes come from? Back in the 60s, a scientist named Lynn Margulis proposed that chloroplasts and mitochondria were at one time independent little bacteria, but they began, probably because they got eaten by other cells, they began to grow inside of those other cells. I know this sounds far-fetched, but this happens all the time. When you get tuberculosis, the bacteria actually are growing inside of your cells. It happens, it's very, very common for something that gets inside of a cell, either accidentally or intentionally, to actually not die and actually grow in there. So it's believed that more than 1 billion years ago, eukaryotes showed up because prokaryotes were eating prokaryotes, and some of the prokaryotes that got ate didn't die, and they lived inside of them. What we now know is mitochondria and chloroplasts have ribosomes, which are protein factories, but they're not eukaryotic-style ribosomes, they're prokaryotic-style ribosomes. And so what I'm trying to tell you is that you have, in a human body, there are maybe 100 billion, or there's trillions of cells. And in each cell, you have mitochondria, which are the little engines that provide the power that make the cells work. Those engines are not yours, actually. They are like tiny bacteria. They grow inside your cells and they keep you alive, but actually their origin is outside of you. So we can't live without them, and actually they can't live without us. It's the ultimate symbiosis. It's the ultimate mutualism. It's the ultimate coevolution on Earth. We talked about coevolution in the evolutionary unit. This is an incredible coevolution where these mitochondria are inside of your cells and they are powering everything that your cells do. The little mitochondria actually have their own bacterial ribosomes. They have their own bacterial DNA. They can reproduce on their own, etc. Trees have both chloroplasts and mitochondria. Chloroplasts are also. They have their own DNA, they have their own bacterial ribosomes, and their DNA is not a chromosome. Instead, it's a hula hoop, just like a bacteria. We call this the endosymbiosis theory, and this was first proposed by Lynn Margulis. It was met with a lot of skepticism at first, and now we, in biology, all believe Lynn nailed it on the head. She was entirely correct. So in the next discussion, I'll talk about organelles inside of cells.